Welcome to this educational video on the classification of chemotherapy agents and their mechanisms of action. Chemotherapy drugs are broadly categorized based on how they kill or inhibit cancer cells. Alkylating agents are among the oldest and most widely used chemotherapy drugs. They work by transferring alkyl groups to DNA bases, primarily guanine at the N7 position. This alkylation causes cross-linking of DNA strands, both within a single strand and between strands, preventing DNA replication and transcription. They also introduce single-strand breaks and base misbearing, leading to mutations. Alkylated DNA becomes difficult for the cell to repair, resulting in accumulation of damage and triggering apoptosis. Alkylating agents are cell cycle nonspecific, but are most effective during the S phase when DNA synthesis occurs. There are four major subtypes of alkylating agents. First, nitrogen mustards, the classic example being cyclophosphamide, which is activated in the liver to form phosphoramide mustard, an active metabolite that alkylates DNA. It is used in lymphomas, breast cancer, and autoimmune diseases such as lupus nephritis. Side effects include myelosuppression and hemorrhagic cystitis. Mesna is co-administered to prevent bladder toxicity. Second, platinum compounds such as cisplatin form platinum DNA adducts that block replication and transcription. They are used in testicular, ovarian, lung, and bladder cancers. Third, nitrosoureas like carmustine cross-link DNA and carbamoylate proteins. They are used for brain tumors and Hodgkin lymphoma. And fourth, alkyl sulfonates like busulfan cause DNA cross-linking and are primarily used in chronic myelogenous leukemia. The second major class is antimetabolites. These drugs mimic natural cellular building blocks such as folate, purines, and pyrimidines. By doing so, they interfere with DNA and RNA synthesis. Unlike alkylating agents, antimetabolites are cell cycle specific. They are most active during the S phase of the cell cycle. They are subclassified into folate antagonists, pyrimidine analogs, and purine analogs. Methotrexate is one of the most commonly used antimetabolites and is a folate antagonist. It inhibits the enzyme dihydrofolate reductase, or DHFR, which converts folic acid to its active form, tetrahydrofolate. Tetrahydrofolate is required for the synthesis of purines and thymidylate, essential components of DNA. Inhibition of DHFR leads to depletion of purine and pyrimidine nucleotides, severely impairing DNA replication and cell division. Clinically, methotrexate is used for leukemia, lymphomas, breast cancer, osteosarcoma, and head and neck cancers. It is also used in autoimmune conditions such as rheumatoid arthritis and psoriasis. Its major side effects include myelosuppression, hepatotoxicity, mucositis, and renal toxicity. High-dose regimens are followed by leucovorin rescue, folinic acid administration, to protect normal cells. 5-fluorouracil, or 5-FU, is one of the most widely used pyrimidine analogs in cancer therapy. It is converted into its active metabolite, 5-fluoro-2'-deoxyuridine monophosphate, known as FDUMP. FDUMP inhibits the enzyme thymidylate synthase, which is required for the synthesis of thymidine, a key pyrimidine nucleotide. Inhibition of thymidylate synthase prevents the synthesis of thymidine, leading to depletion of DNA and RNA synthesis, ultimately causing cancer cell death. 5-FU is commonly used to treat colorectal cancer, breast cancer, gastric cancer, and head and neck cancers. It can also be applied topically for basal cell carcinoma. Side effects include myelosuppression, nausea, vomiting, mucositis, diarrhea, and with long-term use, the characteristic hand-foot syndrome, also known as Palmar-plantar erythrodysesthesia. Purine analogues include mercaptopurine, abbreviated 6-megapixel, and thioguanine, or 6-TG. These drugs mimic the natural purines adenine and guanine. Mercaptopurine is converted into its active form, 6-thioguanine nucleotide or 6-TGN, which incorporates into DNA and RNA, disrupting their synthesis. 6-TGN also inhibits purine biosynthesis, further impeding DNA replication and cell proliferation. Clinically, these agents are primarily used in acute lymphoblastic leukemia, chronic myelogenous leukemia, and inflammatory bowel diseases. Note that allopurinol, used for gout, can increase mercaptopurine toxicity by inhibiting its metabolism, so the dose must be reduced when used together. Side effects include myelosuppression, hepatotoxicity, and gastrointestinal toxicity. Cytarabine, also called arabinesyl cytosine or ARA-C, is a nucleoside analog primarily used in hematologic malignancies. It is converted into its active metabolite, ARA-CTP, which inhibits DNA polymerase and incorporates into the growing DNA strand, causing chain termination. It is especially effective in cells with high rates of DNA synthesis, such as leukemia cells. Cytarabine is primarily used in the treatment of acute myeloid leukemia, non-Hodgkin lymphoma, and chronic myelogenous leukemia. Side effects include myelosuppression, nausea, vomiting, cerebellar toxicity at high doses, and ocular toxicity. Natural products form the third major class. A well-known subgroup is the vinca alkaloids. Vincristine is derived from the periwinkle plant, Catharanthus roseus. It exerts its cytotoxic effects by binding to tubulin, the protein that polymerizes to form microtubules, which are critical components of the cell's cytoskeleton. By binding tubulin, vincristine prevents microtubule polymerization, disrupting formation of the meiotic spindle, a structure essential for cell division. As a result, vincristine blocks cells in the M phase of the cell cycle, preventing proper mitosis, leading to cell cycle arrest and ultimately cell death through apoptosis. It is used in leukemia, lymphomas, and various solid tumors. The dose-limiting side effect is peripheral neuropathy. The fourth class is targeted therapy, specifically anti-angiogenic agents that target VEGF, vascular endothelial growth factor, which is essential for tumor neovascularization. The first subtype is monoclonal antibodies, exemplified by bevacizumab, marketed as Avastin. Bevacizumab is a monoclonal antibody that directly binds VEGF-A, preventing it from binding to its receptors on endothelial cells, thereby inhibiting angiogenesis. It is used in combination chemotherapy for colon cancer, non-small cell lung cancer, glioblastoma, ovarian cancer, and renal cell carcinoma. Side effects include hypertension due to reduced nitric oxide production, wound healing complications, proteinuria, and GI perforation. The second subtype is soluble receptor fusion proteins, such as aflibercept or zoltrap. Aflibercept consists of the extracellular binding domains of VEGF receptor 1 and receptor -2, fused to the Fc portion of human Ig. It binds VEGF-A, VEGF-B, and placental growth factor with very high affinity, acting as a VEGF. The fifth class is topoisomerase inhibitors. Let us begin with topoisomerase 1 inhibitors. Topoisomerase 1 is a nuclear enzyme that relieves torsional stress ahead of the replication fork, by creating a transient single-strand break in DNA and then religating it. Comptothecin analogues such as irinotecan and topotecan work by binding to and stabilizing the complex formed between topoisomerase 1 and DNA. known as the cleavable complex. By preventing religation, these drugs cause persistent single-strand breaks. When the advancing replication fork collides with this trapped complex, the single-strand break is converted into an irreversible double-strand break, triggering apoptosis. These agents are cell cycle specific, most active during the S phase. Irinotecan is widely used in colorectal cancer, while topotecan is used in ovarian and small cell lung cancer. The hallmark side effect of irinotecan is severe diarrhea, along with myelosuppression and nausea. Now let us discuss topoisomerase II inhibitors. Topoisomerase II is essential for resolving DNA supercoils and for chromosome segregation during cell division. It does this by creating transient double-strand breaks, passing another DNA segment through the gap, and then religating the strands. Topoisomerase II inhibitors stabilize the cleavable complex. The enzyme bound covalently to the broken DNA ends, preventing religation and causing persistent double-strand breaks that trigger apoptosis. These drugs are cell cycle specific and most active during late S and G2 phases. There are two major subgroups. First, epipodophytotoxins, derived from the mayapple plant, including etoposide and teniposide. They are used in testicular cancer, small cell lung cancer, lymphomas, and leukemia. Second, the anthracyclines, doxorubicin, daunorubicin, and epirubicin, which additionally intercalate into DNA, further blocking replication and transcription. They are used in breast cancer, leukemia, lymphoma, and sarcoma. The critical side effect of anthracyclines is cumulative, dose-dependent cardiotoxicity, dilated cardiomyopathy. All topoisomerase II inhibitors carry a risk of secondary leukemia. The sixth class is antitumor antibiotics, natural products derived from soil microorganisms, particularly Streptomyces species. They employ multiple mechanisms to kill cancer cells. The anthracyclines, which we touched on earlier, intercalate between DNA base pairs, inhibit topoisomerase II, and generate reactive oxygen species. This combination causes extensive DNA strand breaks and oxidative damage, leading to apoptosis. Doxorubicin is a workhorse agent used in breast cancer, leukemia, and lymphoma, but carries dose-limiting cardiotoxicity. Bleomycin has a unique mechanism. It binds DNA, chelates ferrous iron, and uses molecular oxygen to generate hydroxyl-free radicals that cause single and double-strand DNA scission. It is used in testicular cancer and Hodgkin lymphoma. Its dose-limiting toxicity is pulmonary fibrosis. Actinomycin D, or dactinomycin, intercalates into DNA and physically blocks the movement of RNA polymerase, thereby inhibiting transcription. It is used in Wilms tumor, rhabdomyosarcoma, and Ewing sarcoma. Mitomycin C undergoes bioreductive activation to form a bifunctional alkylating agent that cross-links DNA strands. Used in gastric, bladder, and cervical cancers. The seventh class is tyrosine kinase inhibitors, or TKIs, a cornerstone of modern targeted cancer therapy. Receptor tyrosine kinases are cell surface signaling proteins that, when mutated or overexpressed in cancer, drive uncontrolled cell proliferation, survival, and angiogenesis. TKIs work by competitively blocking the ATP binding pocket of tyrosine kinases, preventing phosphorylation and thus shutting down downstream signaling cascades including the ROS-MAPK and PI3K-ACT-mTOR pathways. The prototype TKI is imatinib, which targets the constitutively active BCR-ABL fusion kinase created by the Philadelphia chromosome translocation in chronic myeloid leukemia. Imatinib revolutionized the treatment of CML and also targets KIT in gastrointestinal stromal tumors. Multi-targeted TKIs like sunitinib and sorafenib inhibit VEGFR, PDGFR, and KIT simultaneously, providing both anti-angiogenic and anti-proliferative effects. They are used in renal cell carcinoma and hepatocellular carcinoma. ALK inhibitors such as crizotinib and alectinib target ALK gene rearrangements found in a subset of non-small cell lung cancers. Class side effects of TKIs include diarrhea, hepatotoxicity, hypertension, skin rash, and QT prolongation. The eighth class is EGF receptor inhibitors, agents that specifically target the epidermal growth factor receptor, also known as EGFR, HER1, or ERBB1. EGFR is overexpressed or mutated in many solid tumors including lung, colorectal, and head and neck cancers, where it drives proliferation, invasion, angiogenesis, and metastasis. There are two main approaches to blocking EGFR signaling. The first approach uses small molecule tyrosine kinase inhibitors that act at the intracellular kinase domain. Gefitinib and erlotinib bind the ATP binding pocket of the EGFR kinase, blocking autophosphorylation and shutting down the downstream ROS-MAPK and PI3K ACT signaling pathways. They are used primarily in EGFR mutant non-small cell lung cancer. The characteristic side effect is an acneiform skin rash, which paradoxically correlates with treatment efficacy. The second approach uses monoclonal antibodies directed at the extracellular ligand binding domain of EGFR. Cetuximab and panitumumab block EGF and TGF-alpha from binding to the receptor, preventing receptor activation, and additionally mediate antibody-dependent cellular cytotoxicity, or ADCC. They are used in ROS wild-type colorectal cancer and head and neck cancers. A closely related target is HER2, or ERBB2, which is overexpressed in about 20% of breast cancers. Trastuzumab, also known as Herceptin, is a monoclonal antibody that blocks HER2 signaling and mediates ADCC, and is the standard of care for HER2-positive breast and gastric cancers. To summarize the complete classification of chemotherapy agents: 1. Alkylating agents including cyclophosphamide, cisplatin, carmustine, and busulfan, which cause DNA cross-linking. 2. Antimetabolites such as methotrexate, 5-fluorouracil, mercaptopurine, and cytarabine, which inhibit DNA and RNA synthesis. 3. Natural products like vincristine that disrupt microtubule dynamics and block mitosis. 4. Anti-angiogenic agents like bevacizumab and aflibercept that inhibit VEGF-driven angiogenesis. 5. Topoisomerase inhibitors. Topoisomerase I inhibitors like irinotecan and topotecan that trap the topo-1 cleavable complex, causing single-strand breaks. And topoisomerase II inhibitors like etoposide and doxorubicin that cause double-strand breaks. 6. Antitumor antibiotics including bleomycin, actinomycin D, and mitomycin C, which work through DNA intercalation, free radical generation, and cross-linking. 7. Tyrosine kinase inhibitors such as imatinib, sunitinib, and crizotinib that block the ATP binding site of receptor tyrosine kinases, shutting down cancer proliferation signals. And 8. EGF receptor inhibitors including gefitinib, cetuximab, and trastuzumab, which target EGFR and HER2 signaling pathways. Together, these 8 classes represent the full arsenal of chemotherapy, each exploiting a unique vulnerability in cancer cell biology. Thank you for watching this comprehensive educational overview.